He is here today. Oops, we're not doing it that way. We're doing it this way.
You can start at kickball.
Yep. And let's see, how do we get this bigger though? Why didn't it go full screen?
Green one.
Green?
It's the green one.
Thank you.
Man.
That's helpful. All right, Riley's here. We're recording. I'm turning off my phone, and this is our last class.
Awesome. All right. Well, good to be back. We're going to talk about more of the clinical side of our T cells today, which I'm excited about. Before, I just wanted to review kind of the whole process because it's been a little while since I've been here. So remember, CAR T cells, we're removing the patient's blood. We then have to isolate the T cells through apheresis. We then have to incorporate the CAR gene into those T cells that we isolated. And we do this through viral transduction. Talk more about that in a second. Really want to nail that home. And we then have to check and make sure that it's being expressed. You can check for protein through Western blots, things like that. Make sure it's on the cell membrane and being expressed in the right ways. And then we want to grow them and then put them back into the patient, their own T cells now edited and programmed to hopefully then be able to kill these cancer cells. Just to touch here again, I just want to talk about how we get the actual CAR gene into these T cells. This is an example using a retrovirus. They noticed that viruses can inject RNA sometimes into cells if they're a retrovirus, and then they can use something called reverse transcription to turn this RNA back into DNA. This DNA can then be brought into the nucleus and incorporated via integrase into the actual own cell's DNA, and then it can be expressed. And in the case of a virus, it's going to go and produce more viruses. But here, they use replication deficient viruses, so they don't go to producing viruses. They just express the gene that you want them to. And so you're not actually producing more viruses. You're just expressing, in this case, the CAR gene. And this is just talking about that again. We have comes into the cell. The RNA comes into the cell. It's reverse transcribed. Integrates it, allows it to get into the genome, and then you can express it. And just some takeaways from last time. Remember that this CAR T cell is modeled after the T cell receptor. It has many parallel pathways that allow for redundancy and flexibility. And that there are these big two kind of responses that happen. One is fast, and it's the non-genomic response. And that was what we talked about with WASP protein and actin dynamics and growth dynamics, which allowed for the immunological synapse to form. It kind of changes the shape of the cell, and that happens really quick. And then there's the genomic responses, which is a lot of what this class is focused on, outside of what I've talked about. These are transcription factors. These are things that are then changing the, pathway that the cell's kind of going. It can totally change what that cell does and its phenotype and everything that's going on with it. And that's the second thing that kind of happens there. Well, it's more cell cycle progression, proliferation, survival. And then hopefully these alterations end up allowing that T cell to go and kill the target cancer cells. So, that review underway. Let's talk about some of the therapies, some of the FDA approved, some of the cancers that have FDA approved CAR T cell therapies. These are, so B cell lymphomas, there's non-Hodgkin's, there's a bunch of different types. Actually, some of these are subtypes of B cell lymphomas. That's not important to know the names, but it does seem to be only working for something called non-solid tumor cancers, which are more blood cancers, leukemias, lymphomas. And so these are just some of the types, just to give you some names. The 5-year survival rate, which is a big kind of thing that cancer studies use to kind of tell how lethal a cancer is going to be, and these are quite high compared to some other cancers, although it's still terrible, the result in any death. It's about 50 to 90% for a variety of these. But of course, that still means that for any one of them, there are still unfortunately patients that are not going to respond to the standard of care. And for B cell lymphomas, the standard of care is generally something that combines 4 different chemotherapy drugs, and it also uses a monoclonal antibody, and this is kind of called R-CHOP. You cycle it every 3 weeks. And then, unfortunately, it's these CAR therapies, even though they are FDA approved, still generally you have to go through the standard of care. You have to be you know, join a wait list. And it has to be recurrent or resistant to that treatment multiple times, basically. And so you're not trying that till the end. Before we talk about some of the benefits, I just want to touch on some of the limitations. Generally, the cost is quite high, and there's a variety of reasons for that we can touch on. But yeah, I mean, $500,000 to $1 million, you know, it can be less, can be more. That's really unfortunate and not feasible for a lot of people. You know, and there's a lot of reasons that could be. It could be because of the issues with production, how personalized it is, how new it is, the fact that, you know, you have to isolate these T cells. There's only certain labs that are able to then edit them and do things. I think that's changing. So as the production speeds up and is optimized, maybe that can come down, hopefully. And there's limited effectiveness. They don't work for a lot of solid tumors. And solid tumors are unfortunately the major cause of death in terms of cancer, like pancreatic cancer, you know, these terrible, terrible forms of cancer. And so it would be great if we could make some headway in that direction. Limited availability is somewhat because of cost, but also because of, like I said, when they're allowed to even try, the patients are allowed to really join a wait list to try these. Are only once it's been reoccurrent, and they kind of get to a point where they've exhausted all their options. So that availability is kind of twofold. And then there's side effects. Let's touch on some of those. So yeah, there's a variety of side effects that can occur. And I just want to kind of compare these to the standard of care side effects, which is a much, much longer list. Neither is great, but, you know, unfortunately, with chemotherapy, it's such a great way to defeat many cancers, but it's not perfect, and everybody understands that. And so you kind of just, it's, you know, you got to pick the poison. And so you have to manage these trade-offs and talk with the patient and their goals of care and what they're willing to sacrifice, and understanding that, you know, CAR T-cell therapy is not a magic bullet by any means, and that there's always going to be side effects. But, you know, the standard of care already has so many side effects that that's, you know, generally not the thing that people are super worried about. So some of the outcomes from some of the trials, there's a lot of jargon in cancer research and clinical research that I just want to avoid. So just to simplify it, for multiple myeloma, there was a 33% complete remission, and in the treatment group, whereas the standard of care therapy group had only a 5% complete remission. So that's pretty great. And then the average time for reoccurrence was still, you know, only a little bit over a year. But there was the average time for recurrence was almost 10 months longer than the standard of care, which is still really great. And I should mention that this study was done in what they call it's triple resistant, so it's multiple chemotherapy resistant. So these are patients who are already, like I said, they are, you know, relapsing. They're not, you know, responding to the standard of care, you know, as one would hope. And so, you know, even though these numbers aren't ginormous, I think that it's still very encouraging that they're on the right track here, and that hopefully, you know, with further research, you can help improve these treatments even more. Just some other statistics. 9 out of 10 people with acute lymphoblastic leukemia didn't respond to other treatments, had full remission with CAR T-cell therapy. That's pretty cool. And of course, you know, whenever I say these statistics, there's still a lot of other people who weren't able to respond, unfortunately. But it is, you know, with these people who aren't able to respond to the normal therapy, it is encouraging to see that there's some other option, potentially. And this part here is pretty cool. For non-Hodgkin's lymphoma and chronic lymphocytic leukemia, which is 70% of complete remission, and about a third of those people had long-term lasting remission, which is something that's very difficult to achieve in people who are continuing to have reoccurrence of their cancer post-treatment. So you see these increased survival rates in complete remission can be long-lasting in some patients. One other two large clinical studies were shown to be more effective than the standard of care for specifically non-Hodgkin's lymphoma after it had reoccurred after one initial therapy. And so that's an important point to think about that I'll touch on a little bit in a little bit, that, you know, perhaps if we shifted this earlier in the treatment regimen, there could potentially be, you know, a greater benefit. And unfortunately, the costs kind of prohibit that right now. And but if it's more effective than the standard treatment for patients, it might have to be a rethinking of when to, start using it. This is another really cool point, is this persistence. CAR T cells, you know, is first done, the first people who they tried it on, it's probably 13, 14 years now. And so they've now detected CAR T cells still in some of those patients up to 10 years later. After their, you know, one infusion, you know, the whole process one time, they can still detect them later. It's not at the same level, but they're still there, which is very good to know. And that like other treatments don't work like that. So, and some of the earliest people treated were over 13 years, are still in remission now. And remember that these people, they didn't have any other options, unfortunately, at that time. And so this for them was really, really incredible. And, you know, they do have side effects, but the side effects are less so than, unfortunately, the shotgun approach that chemotherapy kind of is. And although there's been improvements on that ground, there's still things to work on there. And some of the research I've been working on the past 2 years helps. We're trying to understand the pain that patients get because of chemotherapy, because that can be a rate limiting. Like they can go off of the cancer treatment because of how bad the pain is, and nausea and all the things that come along with it. So there's research on that front trying to combat that. But it's also really encouraging when you have a new therapy that has less side effects, and is hopefully in some cases even more effective. And also this idea of it being personalized is really, really cool. And I think no matter what, even if CAR T cells doesn't end up, you know, making the headway that we all hope, I think this idea of us engineering cells to do a job is here to stay. This idea of, you know, personalized medicine is definitely here to stay, and I think it's a good thing. The new research is definitely focused a ton on these solid tumors, because like I said, this is the big killer. And unfortunately, CAR T cells are not very effective at killing them as they stand right now. And I can touch on two reasons for that. One is because it's very hard to direct T cells to infiltrate certain tissues. It's not very well known how to specifically do that because your T cells are in your blood, right? And they have to then go to the specific distant site of the tumor, and it's very hard to direct them. That's not really well understood. And so getting them there is an issue. And then if they are there and they're like right on the tumor, something happens called T cell exhaustion, which this response is so overactive, and eventually, the tumor is not being killed, and these signalings can get exhausted, and then the response dwindles, unfortunately. Another interesting idea is combining this with other therapies, and this kind of goes along with starting the treatment regimen earlier in combination. And maybe perhaps there's synergistic effects, perhaps, there's unwanted effects, and side effects are even more, but so that's something that people are looking into actively. And perhaps if we were started earlier, we could, that would, I'll go to this side actually. We'll talk more about this last issue. I just want to start at this one here. The demand for CAR T cells is outpacing the supply, right? It's hard, this is a lengthy production process because it's personalized. To take them out from the lab, it can take weeks to do for one person, and it's very costly. If we can optimize this production by perhaps allowing it to start sooner, you can incentivize production and incentivize companies to set up productions that's going to be useful, more condensed, more, just optimized, and allow for things that businesses do well, which is optimize and hopefully reduce those production costs. But then we can, increase accessibility. I think that would be really awesome. And again, the side effects can be serious, but they can be managed. In an earlier CAR T cell therapy will definitely will most likely produce better results. Although that's hard to implement right now on population level. And hopefully there is some leeway here on being able to treat solid tumors, because that would be incredible. That would really be amazing if we could do that. And we don't have to necessarily watch this video. It can be, it's pretty incredible. I think she's got an incredible story. And
I can send out the link with,
Yes.
recording this
Yes.
with that. I can
Yeah.
send out the link to this.
it is only a few minutes, but it is like, I'll just cry. I mean, it's like a beautiful story. And the doctor who helped, it's at CHOPT, it's the Children's Hospital in Philadelphia. There was this patient, Emily Woodhead, and he said, he was asked, is this a cure? Is this a cure? And he's like, it's not, but it is a cure for some. And Emily was about to go on to hospice as a 6-year-old. And the day after they were like, we're not giving up, they happened to open the trial. And, you can see that 2 years later, she's still there. And it's now been, I think, 12 years. And she is doing amazing, hopefully starting college, learning to drive. And it's an incredible story. And, it's amazing. And it's hopeful. And, it's important to remember that it doesn't always still work for other patients, unfortunately. But you gotta, take the wins as they come, and hopefully inspire others to continue. Because, you talk about a home run. I mean, it's incredible. There should just be stadiums, people cheering, the scientists and everybody who helped make that possible. And hopefully where we hope it will go. So.
Thank you, Riley. Awesome. So, question for Riley.
Who are you doing your research with?
I do my research with Dr. Good over at UNE's School of Medicine. She's an immunologist, but they have a big pain center there. They do research into pain, and so the exact research has to do with how the immune system might be modulating and mediating pain response in chemotherapy-induced pain.
Are you guys partnered with other groups that are doing similar research, or not necessarily?
We are actually trying to start a pilot study right now with a local hematology oncologist, looking at their patient's blood in patients with breast cancer, and then correlating that with the pain and the T cells, the phenotype of the certain T cells. And we're just starting that out right now in talks. We're trying to get that approved, so we're trying to do some collaborations.
Has CAR T cell therapy ever been done on non-chemo patients to see if side effects from chemo potentially lead into side effects associated with CAR T cells, or vice versa?
So so it's not always the case that CAR T cells in the trials are done while the patient's on chemo. Usually those chemo spots are in 3 weeks cycles.
Yeah.
And so they do want to deplete it right before, and then the next thing they do is the CAR T.
Right.
So it's not like they're on it.
So my question is has CAR T cell therapy ever been done with patients who haven't gone under chemo? Because chemo is cellular.
I don't think so. I don't think in humans. I'm sure that's been done in mouse models, but I doubt that. Not that I know of, because usually you don't get it till the end. I believe so. I don't think so.
You said we're going to talk about the slide. We're gonna go back.
Which one?
You skip a slide and say we're going to come back to it.
Oh, at this one I said, I'll come back to this on the next slide. Yeah, and then I was talking about the demand, and hopefully that could increase accessibility. Does that make sense?
Is the pain level different for the little girl and the older person?
Is the what level?
Pain?
Yeah.
The pain level? I don't know. I don't know exactly. I don't think so. I think the pain or pain.
The pain.
Pain experience?
Yeah.
Yeah. Well, pain is somewhat subjective, right? And so just speculate and say that, an adult is used to, I'm just gonna stop this chemo, I'm gonna go camping, right? Kids don't have that, right? They're, almost like, a pet animal, right, where they don't have options to take action. So in that sense, probably the perception and experience of the pain is, but then if you don't have action, you just kind of hang in there, I would think.
Yeah, this pain is not so much associated with the CAR T-cell therapy. That pain is more chemotherapy. Like they have something called CIPN, which is chemotherapy-induced peripheral neuropathy, which is just really pain. That's what we look into, right? It's pain in like your fingertips, and it's really, really, really terrible. But that's not so much. I don't think they see that a ton with CAR T-cell.
And lots of pain from the cancer.
Yes, there's cancer-associated pain too. Yeah, but those are usually more solid tumors ones too. But yeah. I should say that Emily was also one of the first, I think she was actually the first kid that they ever tried this therapy on too. And so, people are very hopeful. It's also very important to make sure that, these families and people are like incredibly vulnerable because of the situation that they're in. And so, everybody takes very good care to make sure that people know that this is not, a cure-all. And they're very hopefully educated, whether or not they're able to fully understand that in the moment is hard to say, but that's a lot of care is taken towards that because that is a worry, that people, are unfortunately not, their expectations are different than what the outcomes could be. So that's a hard thing to control for, but it is great when you get to have an awesome outcome like that.
You said that the CAR T cells are still seen 13 years later. Does that mean that they would act in the same way, that they would attack cancer in the same way, or is that something that they're kind of still looking at? Or?
Yeah, that would be the hope. There's only so many people that they have data points on that, right? But that would be the hope, is that if they're persistent, the levels at which they're expressed is much lower, but the hope would be that there's still some activity there, but I'm sure that that's not perfectly known in vivo.
If cancer was to come back for those people, would more production of CAR T cells happen in their body?
That's something that they're actively looking into, because with some people, not everybody has the same level for the same amount of time. If that makes sense, it's kind of person dependent a bit, and the idea is, the hope would be that you would increase that population again, but I don't know if that's specifically been seen. And usually once they notice the cancer's back, it's like, even if those CAR T cells would have hopefully taken care of that and you wouldn't see it. So if you see it again, that's usually a sign that it's no longer working for that cancer that's growing.
So would they be able to get like another infusion or something?
I don't think that they've done like multiple infusions. I think that's, once you've exhausted standard of care, and then now these more, even though some of them are FDA approved, there's more experimental options, then it's an unfortunate situation.
Do you think the cost is going to go down anytime soon?
Well, I'm hopeful. I think, one of the things I kept reading about was that, one, if we get it working in solid tumors, the cost will absolutely come down, because there will be such incentive because of how much of it, that's one of the leading killers. That is the leading killer in cancer. But in terms of like the three largest killers, the reason cancer is so high up is because of the unfortunate deaths that are caused by things like pancreatic cancer, which has like a 1% 5-year survival rate once it progresses to a certain amount. And so the idea is that, if they were to do that, if they were able to optimize towards that, then the production costs would decrease because of how incredible that would be. Another idea is that if you can implement it earlier, you can incentivize production, and you can be able to supply it to more people, and that companies would invest more into producing this super efficiently, because if they can patent a process that does that, that's incentive for them. And unfortunately, these are businesses, so that's something to consider. So it's not a perfect system, but hopefully, if you were able to produce things more efficiently, that would lower cost.
Awesome. Thank you, Riley. And Riley is our tutor too, remember. Our final exam is on Tuesday, and so you might get some students over the weekend or something like that. Godwin, yes. Or no, you're distracted, okay. But yeah, thank you again, Riley, and that was really cool because like, one of the learning concepts in our course is novelty. The novelty of, oh, we worked with this, clay, or we learned about this, or we heard this from Riley, right? So when we think CAR T cells, we can think about Riley, and we can cluster all our knowledge about CAR T cells, focus it on Riley. And we were, oh, that Riley caught that guy, right? And those are effective learning strategies, and so that's part of it. Then another part of it is let me just get this going.